Hello everyone, and thank you for having me here today presenting at the LabRoots Neuroscience virtual event. My name is Gemma Gomez Giro, and I will be talking to you today about patient-specific brain organoids for in vitro modeling of Parkinson's disease. First of all, I would like to introduce myself a bit more. I come from Spain, from Barcelona, where I did my bachelor and my master's degree in biomedical sciences, which is my background. Then I moved to Germany for my PhD, which was involving stem cell biology, neuroscience, and neurodevelopment. And this was my first contact with stem cells, 3D cultures, and disease modeling as well, using these technologies. And after that, I did a postdoc at the University of Luxembourg and the Luxembourg Center for Systems Biomedicine, involving also organoids and the development of on-chip technologies to study Parkinson's disease with these organoids. And since October last year, I am a project lead scientist at OrganoTherapeutics, which is a spin-off from the University of Luxembourg. And in this talk today, I will talk to you about Parkinson's disease and its main pathological features. I will introduce the concept of organoids as well as assembloids and explain how they are applied to the study of complex neurodegenerative diseases such as Parkinson's disease. But last but not least, I will also give you some examples about how we use these models to test compounds as potential treatments, especially for targeting Parkinson's disease. So Parkinson's disease is the second most common neurodegenerative disorder worldwide after Alzheimer's disease, affecting about 10 million patients. And even though a lot of effort has been placed into developing therapies, all treatments currently available are directed to managing the symptoms, and no curative treatment is available. Therefore, patient care is another big pillar in disease management, contributing to the total economic burden of the disease, which is estimated to be around $52 billion per year, according to a study published by the Michael J. Fox Foundation only in the US. Consequently, there is a lot of interest in developing new and better therapeutical approaches for these patients. Regarding its clinical manifestation, the most characteristic sign of PD or Parkinson's disease is the presence of motor symptoms, which include tremor and postural instability. But patients also present a variety of non-motor symptoms, including sleep disturbances or loss of smell, which can actually appear a long time before the motor symptoms arise. The cause of these symptoms is the loss of a specific neuronal population, the dopaminergic neurons, in a very specific region of the brain, which is the substantia nigra in the midbrain, whose name is attributed to the presence of dark pigment called neuromelanin in these areas. You can visualize what I just said in the cross-section of the midbrain that is in the slide, which compares the presence of these neurons and the pigment in the healthy individuals as compared to their loss in the brains of PD patients. And the problem is not solely the death of these neurons in the midbrain, but that these neurons are actually connected to other regions of the brain, such as the striatum. And the depletion in the striatum of dopamine, for example, causes also pathological symptoms in Parkinson's disease. Parkinson's disease is a multifactorial disease, and this means there are different factors that contribute to its development. Some are genetic, with several genes that can drive the manifestation of the disease or increase the risk, but these cases only account for 5 to 10% of the cases, whereas 90% of the cases are actually sporadic. Age is the biggest risk factor for developing Parkinson's disease, which goes along with the increasing in life expectancy of the populations. Among other contributing factors are environmental factors, such as the use of pesticides or industrial toxins, as the incidence of PD is more frequent in industrialized countries. But Also, lifestyle is a big contributor, such as, for example, the lack of activity or the diet, or comorbidities with other diseases such as type 2 diabetes or brain injury can also contribute to the disease. As I mentioned earlier, most patients at the time of diagnosis already present with motor symptoms, and this happens already when 40 to 60% of the neurons in the substantia nigra are lost. But the disease can actually start even 20 years earlier with non-motor symptoms, which are constipation or sleep problems, for example. But what we are actually interested in is in going even farther back and study what are the earliest neuronal changes that can lead to Parkinson's disease. And this is with the idea that Parkinson's disease presents a developmental component. We believe that there are neurodevelopmental defects already happening at the embryonic stage, probably associated with mutation in the PD-associated genes, which contribute to the first hit to developing Parkinson's disease later on in life. However, a second hit happening later in life and probably correlated with other factors need to happen to actually trigger the disease. The classical models to study PD are animal models, and they have improved greatly our understanding of the disease and the mechanisms involved in the pathology. However, these models do not always recapitulate the pathological hallmarks of the disease or its progression in humans, and the use of these models in the lab to study diseases are posing more and more ethical concerns. Another model much used to study Parkinson's disease is the adult tissue. However, since one cannot simply take a piece of the brain to study it or reach certain affected brain areas, we use mostly postmortem samples, which means, yes, we can look at the actual tissue and at the specific affected brain areas, but it also means that we are looking only at the end stage of the disease. Besides, the quality of these samples, considering that we need to preserve them quickly to be able to study them, can be affected. So the problem is that we cannot study such a complex disease by just looking at a single gene or molecule. And as I mentioned earlier, models that are not human do not recapitulate fully the disease happening in humans. And they also don't consider sufficiently the differences that are present between individuals, but also between patients themselves. Therefore, there is the need to develop more complex human models to study the disease and to study it at earlier stages. To facilitate this, about 10 years ago now, Yamanaka and colleagues developed the technology by which fibroblasts, which can be isolated from a skin sample, can be reprogrammed into induced pluripotent stem cells, or iPSCs, from which we can derive most cell types of the body, allowing us to study the specific cells involved in the disease, be it from healthy individuals or patients, so we can compare them. So we have a tool now that allows us to actually derive the cell type affected in Parkinson's disease in vitro and compare it between patients and healthy individuals. And even though we can do that, dopaminergic neurons alone are not isolated inside the brain, but there are other multiple cell types which compose the brain and contribute to the overall pathology, meaning that the physiological relevance of these models, 2D models in a dish, can be improved. And therefore, three-dimensional culture models have been developed to increase the complexity of the tissues in culture in an effort to bring them closer to the actual body physiology. In this regard, organoids came out as a newer and more complex models to study diseases. Organoids, as such, are three-dimensional, self-assembled cellular structures which are generated from stem cells, which mimic the structure and the functions of the organs they model. And with the years of developing this technology, there are different models for different organs that have come out, such as organoids for the brain, the lung, or the intestine. And within the brain organoids, there have also been a lot of effort to develop not only organoids that resemble the whole brain, but region-specific organoids to study precisely that specific regions of the brain. And in this regard, we focus on midbrain organoids, which are the organoids modeling the substantia nigra of the midbrain, which is the region affected in Parkinson's disease. So how the workflow works is we start with patient or healthy cell samples that were reprogrammed into iPSCs, and we further differentiate those into neuronal precursor cells, which are still stem cells, but they are already committed to the neuroectoderm. And these are the cells we use as a starting population for generating our organoids. And how it works in a dish is we basically put a certain amount of these cells into U-bottom shaped ultra-low adhesion plates and let them self-assemble into spheres. And we then guide their differentiation and maturation as organoids. In between, depending on the different protocols and the later on applications, we sometimes give them some extracellular matrix for support. And then we study these organoids at different stages of cultures, since depending on the culture length, the organoids present different properties and also different cell types. And we use them, as I mentioned, to study the disease in vitro in a patient-specific manner. I will show you first that these organoids actually present midbrain identity. And we know that because we follow the expression of markers from the development of the midbrain, such as FOXA2 or LMX1, and TH for dopaminergic neurons that you see here in the slides. But these organoids also show different cell types, as I mentioned before, that are normal for the region of the brain, such as the presence of astrocytes and oligodendrocytes, which is highlighted here by the markers like GFAP or S100 beta for astrocytes or OLIG4 for oligodendrocytes. These organoids do not only show the cells that are present in the midbrain, but also present functional characteristics like synapse formation or dopamine production and release, which is the actual neurotransmitter released by dopaminergic neurons. They also show synaptic activity through spontaneous firing when we place them on the multi-electrode array, and they also contain neuromelanin, which, as I mentioned at the beginning, is this dark pigment present in the neurons from the substantia nigra. To confirm the identity of the midbrain organoids, we compare their gene expression signature to data sets available from the human midbrain. And as you can see here in the graph, where the different cell populations are highlighted by the different colors, we can see that the midbrain organoids present the same populations which are present in the actual human midbrain. This shows that midbrain organoids can be used in vitro as models to study diseases affecting the midbrain. And what's more, these organoids resemble the developing midbrain, correlating closely with developmental weeks 9 and 10 in the embryo. Therefore, they are a great tool to study early events related to PD. And in the following slides, I will show you these. So one of the key characteristics I've been talking about for Parkinson's disease is the loss of the dopaminergic neurons in the substantia nigra. So we can check in our closest model of the substantia nigra, which is the midbrain organoids, if these neurons are actually lost. And we can see in the graphs two things. One is that we can see the loss of dopaminergic neurons in the midbrain organoids that were generated from PD patients carrying a mutation in the LRRK2 gene. And second, that we are only able to see it when these neurons are actually cultured three-dimensionally within the organoids, but not in the case of dopaminergic neurons differentiated in 2D from the same patient. Characteristic of Parkinson's disease is the presence of cellular aggregates, which are composed to a high extent by a protein called alpha-synuclein. And what we can see in the images is that we can observe these aggregates, which are highlighted in red, in the midbrain organoids that are derived from patients who carry a mutation in the synuclein gene. But we cannot only visualize these aggregates, but we are also working together with collaborators on developing and establishing new tools to assess the presence of these protein aggregates in our organoids. In these graphs, for example, the increase in fluorescence should be proportional to the amount of aggregates which is present in the samples. But one limitation of the midbrain organoids as such is the lack of other components which would be normally present in the brain, but that they cannot be differentiated together in culture in the dish with the organoids, because, as I mentioned before, we use pre-committed cells to generate these organoids, and these missing cell types are arising from different germ layers in the embryo. And I am referring here to components such as the vasculature or the immune cells of the brain, the microglia. Having organoids containing these cell types would help us to study key features of the diseases as well, since the immune system, for example, is very involved in the pathophysiology of PD. Therefore, we are working on making these models even more complex by generating what we call assembloids. And assembloids can be referred to as the addition of or incorporation of other cell types into one organoid, such as the microglia or endothelial cells for vasculature, or as well as the fusion of two organoids, such as a midbrain organoid together with a vascular organoid, or different region-specific organoids, such as a midbrain and a striatum organoid, or a midbrain and a hindbrain organoid. These assembloids are another tool to study the complex interplay between different regions within the brain, but also between different organs on the dish. So we work on the establishment of a protocol to incorporate these microglia, the immune cells of the brain, into our midbrain organoids by generating independently macrophage precursors from iPSCs and incorporating them later on to the organoids. And you can see this incorporation of microglial cells in green into the organoids in the images, which is positive for IBA1, which is a microglial marker. And using this new assembloid model, we can study the neuroinflammatory component in PD, which with midbrain organoids alone we could not study before. And because neuroinflammation is also present in PD brains, and here we can see that in our cultures, we can detect the increase of pro-inflammatory cytokines in the case of midbrain microglia assembloids that were derived from PD patients with both mutations, with two different mutations in the LARC2 gene or the synuclein gene. And with all this, until now, I have showed you that the midbrain organoids and assembloids are a valid tool for studying PD because they recapitulate key features of the disease. But let me show you a few examples of how we can use this model to test the effect of treatments. For example, here we have the case of a PD patient with a specific mutation in the DJ1 gene, and from which midbrain organoids were derived and treated with two different drugs. And you can see both in the images and in the graph how upon 25 days of treatment, the dopaminergic neuronal loss that was observed in the conditions that were untreated can be rescued with the treatment. And not only that, but there is an ongoing collaboration with the Technical University in Munich, in which 25 patients, which are enrolled in a clinical trial for the drug Fasudil, will be treated And at the same time, organoids from the same patients will be developed and treated as well in order to make a correlation of clinical data in the patients and experimental data from the organoids, running this clinical trial in parallel in the patients, with the patients and in the lab at the same time. And this highlights the use of organoids to support the decision for treatment options, entering into true personalized medicine scenarios for PD. Another example is a treatment performed in midbrain organoids aimed to decrease the levels of the aggregated alpha-synuclein protein using antisense oligonucleotides. As you can see in the images, upon treatment, these aggregates of alpha-synuclein in the midbrain organoid sections decreased. And like that, there are numerous publications which support the applicability of patient-specific midbrain organoids for both in vitro modeling of Parkinson's disease, but also for drug testing and development. So, all in all, I hope I could convey the following taking messages about what are brain organoids and the concept of three-dimensional cellular structures that mimic the early brain, that we can make them patient-specific thanks to the availability of stem cells. And I hope I could convince you that specifically midbrain organoids can recapitulate in vitro the main features of Parkinson's disease pathology. And I also showed you how we can use them to test and develop compounds against different forms of the disease in an effort to make the treatment personalized. And with that, I would like to introduce and thank the team here at OrganoTherapeutics. And I would also like to thank the organization of this event and my funding, and all of you for your attention. Thank you very much.